Well, this is going to be a little different if you watch the Bristol Myers deep dive. It's going to be a little different from that because when you look at a company like Bristol Myers, you're kind of doing this like portfolio evaluation of quite a number of different assets in one kind of like bucket. And that's a very different investment proposition from looking at a single asset company or a company with 1 or 2 or 3 assets. When you look at a company like that, you really have to dig deep, really, really deep. It can't be superficial. It has to be this really insane, deep dive. And you can't leave details for just anything. So in any event, let's sort of look up the model. So anyway, this company has Amtagvi, which is lifileucel for melanoma. And it's cell therapy. I've always hated cell therapy. The reason is change is scary to humans in general. We always get scared of new things. But change is often a really good thing. We have to think about changing our mental models and our frameworks for the way we look at things. One of the things that is scary about cell therapy is it's very different from the way we're used to looking at medicine. We're used to looking at medicine as this is a molecular structure that I can rest easy on, right? I mean, one of my favorite drugs of all time is called Plavix. It's also known as clopidogrel. I'll show you the molecular structure real quick. Plavix was a best-selling drug, sold many billions of dollars, saved many lives. So one of the first great blood thinners. And very easy synthesis, very simple molecule, not very many atoms in fact. You have this heterocycle, benzene, 1 chlorine hanging off there, hydrogen coming out of space here, and a simple functional group there, and you have a great drug. And then when we went to antibodies, the molecules were a lot bigger, but they were still sort of familiar in the sense that amino acids make up these gigantic proteins. So that's okay. They're very different, but they're large, and it took a little getting used to, but that was fine. Cell therapy is totally different. Cell therapy is this idea that you can take actual full cells and put them into people as medicine. Now, cell therapy wasn't new, interestingly. We've been doing autologous stem cell transplant, and allogeneic stem cell transplant for a long time. But why are cells so strange? Well, one, it's hard to characterize them. They're kind of a black box. We really don't understand what's going on in a cell in perfect detail. We understand the broad strokes for sure, but that's one big question. So what the heck is this cell doing? The other is just from simply the source perspective and manufacturing. Where are these cells coming from? And we know that if we give somebody else's cells into your bloodstream, your immune system is going to have a really potent reaction to that. And that's why with stem cell transplant, we eradicate the immune system and try to get a graft, and you still could get graft versus host. That's the sort of question that's very important. Where are these cells coming from? Maybe they're coming from the patient, maybe they're coming from somebody else. And the last thing that really bothers any traditional drug person about cell therapy is pharmacokinetics. In drugs, we can measure how long the drug stays in the bloodstream, and we can do that in animals. And we can use something called allometric scaling to predict how it'll work in humans. And when a drug has good pharmacokinetics, or PK for short, we can advance it further into human clinical trials. If it doesn't, we can't. There's no way to tell with cell therapy kind of where is the drug, quote unquote, because it's cells, not a drug. Where are these cells in the body? Can we trace them? Can we track them? Not really. We just sort of hope that they kind of engraft or stay or something. And that's why a lot of these cell therapies are very akin to stem cell transplants. They're not exactly, the ones that aren't sort of transplantable don't do very well. So let's look a deep dive into Amtagvi. We have a lot to learn about this drug. Pretty clearly here it says, your melanoma, your cells, your treatment. Okay, well that seems reasonable. When you get prescribed a medicine, you don't have to figure out where to get it from. Any pharmacy, something like that would work fine. But here, you really have to go to a special treatment center, which is, again, very, very odd. That's because the drug is going to be manufactured just for you, which is really wild. Again, crazy idea. One of the big questions I have here is what kind of conditioning, if the patient gets any conditioning, what kind of conditioning will the patient get? And we're going to look into that. I think I looked into it last time. Lymphodepletion with IL-2. So this is a very light conditioning regimen. Usually when you do a conditioning regimen, we're looking at a company called Iovance. Trades on NASDAQ. It's a $11 stock, 300 million shares outstanding, so around a 3.2 billion market cap, 400 million in cash, so 2.8 billion enterprise value. They have an FDA-approved drug called Amtagvi. So it looks like you take, and kind of the best way to like really do a deep dive and research this stuff, sorry, is going here. So we're going to type in 𝗕𝗏𝗚𝗚𝗚𝗏. Hope I spelled that right. Great. And you can see there's 16 papers, one in JCO, which is a well-known journal. Let's see if there's another well-known journal. Usually it'll be at the top. All right, well, at first glance, that's one paper of interest. But typically, you would want to read every single paper that gets published about this drug. So let's start with this one. JCO is a pretty good journal. Oh yeah, the pharma book I was talking about was was a Merck chemist. My friend was mentioned. Many of my friends were mentioned in the book. Actually, really it was a Merck guy. All right, well I think I have to give up because I'm getting too sidetracked. So let's talk back about Iovance here. All right, so they call this a tumor infiltrating lymphocyte therapy. To me, those words don't mean anything. That's really not that specific. I don't glean anything from that. I just call that cell therapy. So I want to see the details before I'm willing to think about that further. Who are the authors is one of the things I look at. I recognize some of these names off the bat, but let me just see who the first author is. Okay, this Ahmad Sarnak. It looks like he's, see, it doesn't really say what his affiliation is, which is not great. Oh, there we go. He's at Moffitt. So most of these people are clinical investigators that have worked at, that work at different cancer centers. In fact, it looks like maybe all of them are. Yeah, I recognized Chesney, who's a well-known physician. Oh, there's one or two guys from the company, Iovance. I see their names there. Let's recognize some of them as well. All right, so typical clinical paper here. Effective treatment options are limited for patients with advanced melanoma who progress after checkpoint inhibitors like Keytruda and targeted therapies. Adoptive cell therapy doesn't mean much. Cell therapy using TILs, or tumor infiltrating lymphocytes, not really, again, a meaningful term. So I would just say cell therapy has demonstrated efficacy in advanced melanoma. 𝗕𝗏𝗚𝗚𝗏 is an autologous. Aha, now we know what that means. It comes from the patient. Centrally manufactured, so it's not manufactured locally or whatever. Tumor infiltrating lymphocyte product. Again, what's a lymphocyte? You know, that's a broad class of white blood cells. It's not really clear what that is. Is it T cells, for example? So everything here so far is understandable, right? And a lot of these words don't mean anything. They're words that are used to, I mean, I hate to say it, but like a lot of scientific writing, the goal of communication is to be exact and to convey the precise context you wish to convey. In scientific writing, a lot of times there's just a lot of nonsense. But so far we know what the drug's called, lifileucel, maybe. And its goal is to treat melanoma, and it's a cell therapy. So it says here, we conducted a phase 2 open label. What does that mean? Single arm, meaning everyone got the same thing. Multi-center, meaning more than one hospital. Study in patients with advanced melanoma. So open label basically just means there's no blinding. Everyone knows exactly what the protocol is going to be. And so these were patients that had previously been treated with checkpoint inhibitors and BRAF/MEK inhibitors. So these are really bad patients. They are very, very sick. They are likely to be dead. How do you know that? Well, you can do some research on that yourself. You know, people who have metastatic melanoma and they've no longer responding to a checkpoint inhibitor, and they're no longer responding to these drugs, which don't work very well anyway, you have a very, very limited lifespan there. Now, there is one thing you have to ask every time you look at a medicine is, has anything else happened for this disease? And the answer is yes. And in fact, Bristol Myers, which we looked at last time, has a drug for metastatic melanoma. And it's a brand new antibody. Let's see if we can pull it up. That's why, like I said, it's handy to specialize in a sector because you end up kind of getting insights from one thing to the next. And it doesn't look like we made a page for it, but 𝗕𝗏𝗚𝗑𝗢 is the drug I'm talking about. It's a LAG-3 antibody, and they combine it with the checkpoint inhibitor. From a competitive perspective, but also from a scientific integrity perspective, it's interesting that this drug was studied before the 𝗕𝗏𝗚𝗑𝗢 era. And that is important because it sucks to study a medicine while another medicine is changing the game for that disease. Kind of your medicine is maybe a little less relevant now, because people are going in that other direction. So it's a very, very interesting way to look at it. Anyway, so these are very, very sick people. Metastatic melanoma doesn't sound bad. It's like, oh, melanoma, how bad can it be? It's skin cancer. Most skin cancers are not bad, like squamous cell carcinoma and some of the other basal cell carcinoma. But metastatic melanoma means the melanoma has spread from your skin to a vital organ, and it's a very aggressive disease typically. So it says here, lifileucel was produced from harvested tumor specimens. So they harvest your tumor in central CGMP or central GMP facilities using a streamlined 22-day process. Okay, so it takes 22 days. They cut the tumor out of you, or part of it. They cut all of it out of you, but it's on a vital organ, so they can't cut it out of you. So they cut what they can out of you, send it to the facility. It takes 22 days, and then they send it back. So this is very interesting. So typically when you send cells back, imagine sending somebody 1 million cells. Well, how many cells are in the body? Trillions, right? So how many blood cells are there in the entire blood system? Number of lymphocytes. We do know this from complete blood count, right? If anybody went to medical school, 1,000 to 5,000 lymphocytes in a microliter of blood. In fact, I have a CBC here somewhere. And if you have more lymphocytes if you have an infection than less if you don't. If you have those lymphocytes for a microliter, we know there's 5 liters of blood, right? So let's see. So there's 5 million microliters. And if there's 1,000 on the low end, to 4,800 on the high end, there's going to be 5 billion lymphocytes, or on the high end, 24 billion lymphocytes. So if I give you 1 million cells, is this really going to make a difference? No. They could be super cells and they still won't do anything, right? It's just not a lot. It's 1/5000th of the amount of cells you have. Well, if I give you another 0, let's say 10 million cells, that's still not very many. How about 100 million cells? Well, maybe that's starting to get there. That's 2% of your lymphocytes, maybe. We're going to try to replace the lymphocytes, but the problem is we can't replace the existing cells. Or can we? Well, it says here that patients received a non-myeloablative lymphodepletion regimen. So typically with these cell therapies, especially for transplant, you want to wipe out all of the cells. You want to wipe out the whole bone marrow, the whole blood cell kind of system. Excluding the red blood cells. And you can do that with chemotherapy. And in fact, if you ever get a stem cell transplant, the reason it's so tough is you're going to be in the hospital with no immune system. And you're put into almost like a clean room so you don't get an infection. And that is a very, very, very tough process. In fact, it can be lethal. The treatment can be lethal, which is really frustrating for physicians. I'm trying to save your life, but the therapy has a good chance of killing you. And that's why stem cell transplant is a last-ditch effort often for very, very severe diseases. It's not done lightly. So myeloablation, ablation means to destroy, right? Ablative, ablation. We even are now starting to use this word in AI, believe it or not. But it just means to destroy or remove or something like that. And so myelo is the bone. Well, myelo is really the bone marrow, but myeloablative is what it sounds like. So typically you get this myeloablative regimen of chemo. And so that's not what they're doing here. Interestingly, they're doing something called lymphodepletion, which again is not really a word that means anything. What does that mean? They're going to try to lower your lymphocytes. Well, that sounds like myeloablation euphemism to me. They're going to lower something with IL-2. IL-2 is an old drug. In fact, if you know the history of biotech, it was maybe the first, I want to say it was the first cytokine ever cloned and made under recombinant DNA technology. It was made by a company called Chiron, CHIRON, which was a legendary biopharma company that did a lot of firsts. I want to say it was in maybe 82, but I don't remember exactly. I wasn't born yet. So it says here that they got the regimen, which we know is IL-2, but they don't say that here yet. And they got a single infusion of the drug. So you get a single infusion. Administer single infusion after lymphodepletion. And then you get 6 doses of high-dose IL-2. Is that for more lymphodepletion? I don't get it. 6 doses of high-dose IL-2. Okay, so we'll get the deep dive on the regimen exactly. Sorry, I don't do deep dives unless you're a Godel customer. I appreciate all the Godel customers, and even there I don't have the time to do deep dives for everyone. Godel is a terminal software me and my colleagues make that we think is going to compete with Bloomberg and many other great terminal software companies. We have a lot of fresh ideas. I think a lot of the people out there that are trying to make financial software aren't doing it in a intelligent way. They don't really understand what traders really want. I think we do, because we are traders. We traded our whole lives. So the primary endpoint, and this is really important, in every clinical trial there's a primary endpoint. And I was telling a friend the other day about Bonferroni. Oh, I should get his name right. Bonferroni, I believe. Yeah, I got it. Bonferroni correction. And this idea that multiple comparisons are invalid in statistics and inference. And it makes sense. If you played a basketball game, and we know what the primary endpoint of a basketball game is, right? A primary endpoint of the basketball game is points. Whoever scores the most points at the end of the game wins. It's not at halftime. It's not how many assists there are. It's not how many slam dunks there are or three-pointers. In basketball, we know what the primary endpoint is ahead of time, and we don't change it. So the primary endpoint in this clinical trial is something called ORR, or objective response rate. And there is a literal handbook on how to measure this, and it basically means tumor shrinkage. So a response is a certain amount of tumor shrinkage, and you typically need 50% tumor shrinkage. And they talk about what exactly, how exactly you measure by the axis, the long or the wide, and sometimes these tumors are pretty hard to measure. So, you typically need a 50% reduction from the size. And for a complete response, which is obviously pretty awesome, you want a complete disappearance of the tumor. Getting a complete response is awesome in cancer. It's very rare, but a complete response means the tumor's gone. A partial response means 50% or more of the tumor's gone. So that's kind of how that works. So the primary endpoint here is objective response rate. And again, I can't stress how important this is, and I want you to wrap your mind around what statistics is like. If in the middle of the basketball game we said, the winner is going to be determined by how many 3-points there are, and it just so happens that your team is leading in 3-pointers, but is down in total points. Well, you say, well, that's not fair. You can't change it in the middle and then say, oh, well, we did so well with 3 points, but we missed the game because we lost by points, but we won by 3 points. And it sounds silly when you do it in basketball, but when you do it in medicine, people do it all the time, and nobody says it's silly, but it is. And smart people who know medicine know how invalid it is to do something like that. Now, we can note what happened, but we can't take it as seriously as the pre-specified primary endpoint, meaning before the trial started, we said, this is what we're going to measure this drug's by, this successive drug, this drug. We're not going to look at anything other than that from a statistical perspective. We might note what happens, but the main goal has to be set before. It's called a priori sometimes, or just pre-specified primary endpoint. Okay, so it says here 66 patients received drug. There's a bunch of different numbers here from different cuts of this data, but let's just type N equals 66 with 3.3 prior therapies. That's a mean. So that means that the average patient had a little more than 3 prior drugs, which is a lot for melanoma. So they probably got some chemo. They definitely got Keytruda first. Then they got another PD-1 inhibitor. then they may have gotten a targeted therapy and then maybe some chemo, and none of that worked, and they're dying. And these are people who are very, very sick. 100% of them got a PD-1. A number of them got CTLA-4, and a number of them got a BRAF drug. And so what they found was 36% of patients had an overall response, which is kind of good. So a third, roughly 36% of patients had their tumor shrink by 50%. And imagine yourself as one of these patients. You have metastatic melanoma, maybe it's spread to your liver, and your doctor says you have 6 months to live. You take Keytruda, and that keeps you alive for 6 months. Then you try CTLA-4, it doesn't really work. Then you try a MEK inhibitor, it doesn't really work. And you may have a few months to live at this point. And then you try this drug. All of a sudden, your tumor shrinks. But you only have a 1/3 chance of your tumor shrinking. But it doesn't disappear completely. It may only shrink by 50% or more. But it also is important for the other 64% of people whose tumors didn't shrink by 50% or more. What happened to their tumor? Because if it just stayed in check, well, that's better than the tumor growing or you dying. And certainly of these 36% of people who were lucky and had their tumor shrink by 50% or more is a pretty good thing. So 36% ORR with 2 complete responses. So 2 patients had complete disappearance of their tumor, and 22% had partial responses. That's the 2 people was quite good, but that's only 2 out of 66, which is about 3%, right? So you have a 3% chance of a cure. But you have a 36% chance or 34% chance, 33% chance of a 50% or better tumor reduction. And you're not going to die if your tumors are shrinking, which is great. Disease control rate is sometimes what we call people who don't progress, but their tumors don't quite meet the objective response. They don't shrink by 50%, they might shrink by 20% or something like that. So 80% disease control rate means, well, the tumors may not be shrinking by 50% or more, but they're not progressing. The other 20%, unfortunately, did progress and most likely passed away. The median duration of response. So if we just take these 36% that do respond, how long do they respond for? Because it's no good to me if my tumors shrink for 2 weeks and then they come roaring back, and that does happen sometimes. In this case, it looks like the median duration of response was at least 18.7 months on it. Well, that's the median. But it does look like some of those patients had a response. You can see this one for 0.2 months. Well, what is 0.2 months? That's really not very much. That's 6 days before the tumor came roaring back. But on average, if you do respond to this drug in that 36%, you will have a decent response. In the primary refractory to PD-1 subset, the ORR and disease control rate was 41 and 81. Now, I thought all of these people were refractory. It's at 100%, so I'm a little confused. Maybe it meant that they were completely refractory to PD-1. I think that's what it means. Not that they took it, it worked okay, and then they progressed, but from the start, PD-1 never worked for them. I'm not sure this is even worth noting, but I'll write it down anyway. Refractory cohort, 41% ORR, 81% disease control rate. Safety profile is consistent with lymphodepletion and IL-2. It doesn't sound like there are any side effects from the therapy, but these are very sick people, so there is a tolerance for side effects. Now, that's the summary of the article. We're going to start reading the actual detailed article, and we do need to do this. I know there are a lot of people that have ADHD that don't want to do all this work, but I hope to show you that you do have to do the work. All right, so indication, of course, is metastatic melanoma. The treatment of metastatic melanoma with immune checkpoint inhibitors, that's Keytruda and Opdivo, and targeted inhibition with BRAF and MEK has improved patient outcomes. 40 to 65% of patients with advanced melanoma have primary resistance. Okay, so that's what they're sort of saying here. 40 to 65% of those with initial disease control, 30 to 40% develop an acquired resistance. Usually that means some kind of mutation. And so 15 to 20% of BRAF's V600, V is the amino acid called valine, and 600 is literally the 600th amino acid. So BRAF is the gene, and the 600th amino acid in this gene is valine, and it moves to some other amino acid, which is called a mutation. not that hard to read these things if you know what you're reading. 15 to 20% fail to respond to targeted therapy, and only 22% remain progression-free at 3 years. So very, very tough disease. Although primary resistance is lower in patients treated with PD-1 antibody plus CTLA-4 antibody, those are 2 drugs together, and they're fairly toxic. These targeted therapies that promise to eliminate the side effects of chemo, well, we don't quite get there yet. 36% of, and I understand PD-1 is not exactly a targeted therapy, but it was supposed to be this post-chemo era. It's just a different kind of chemo in a lot of ways. 36% of patients discontinue therapy because of adverse events. That's what I'm talking about. 88% because develop immune-related adverse events. These drugs work by revving up your immune system, the PD-1 inhibitors. So anyway, patients progressing after PD-1 and CTLA-4, and after targeted agents have very limited options. 4 to 10% have responses to chemo. And the limited median overall survival is 7 months. I said 6 months earlier, so not too bad. It's gotten a little better, I guess, but it always used to be 6 months. So now you have 7 months if you failed all those drugs. So really dire prognosis. There are no treatment options with approval based on data from patients with melanoma who have progressed after checkpoints for BRAF wild type. Wild type just means the normal mutation, or not the normal mutation, the normal gene that everyone has. In other words, the gene that's in the wild, hence wild type. For one line of, so these would be second line patients who have failed Keytruda or Opdivo, or 2-line for people who have failed Keytruda or Opdivo, and also a BRAF specific drug that specifically targets that mutation. In addition, patients recurring with melanoma after adjuvant, so this is before surgery, PD-1 therapy for high-risk disease represent emerging unmet need. Great. So this is just a background of basic melanoma background. And that's what's nice about these papers is usually the first page will give you a nice, soft introduction to the topic. Sometimes these papers just dive right into the deep science, and if you're new to the space, sometimes when I read papers in other disciplines, I get lost, within the first sentence, and I don't like that. So unfortunately, that's how it works. All right, adoptive cell therapy with TIL, which again, I think is their term, which I don't like. Everyone wants to make a euphemism to stand out, but it's basically, this whole thing is just a cell therapy to me. Cell therapy offers a potential therapeutic option for melanoma, but it has not been studied extensively in the checkpoint era, or the PD-1 era. Cool. Their cell therapy, which they call TIL, are enriched with polyclonal T cells with diverse antigen specificity. And again, I kind of want to understand how they get there. Extraction of a fragment of tumor followed by ex vivo expansion. So expansion means letting the cell divide and growing the cells. So you have to feed the cells, and you have to put them in a place where they're able to grow. So autologous, let's see, tumor resection followed by expansion. So ex vivo expansion means outside of the body. Vivo means life, or in vivo means in life. Ex vivo means, well, we're going to take the tumor out of you and then we're going to put it in some kind of equipment and expand it there. So extraction of a fragment of tumor followed by ex vivo expansion removes TIL from the hostile tumor microenvironment and reduces the immunosuppressive effects of intratumoral regulatory T cells. Let's break that down a little bit more. And I firmly believe you can understand anything if you slow down enough. If you get frustrated and angry that you don't understand anything, well, you're never going to make it with that perspective. But if you slow down a little bit and look at it, it's not that hard. So we got this part, extraction of a fragment of tumor. That's pretty simple, snip, snip. Or it's actually like a punch, hole punch sometimes. But I think this is actually more of a surgical knife resection. So it's a very serious one. All right, followed by ex vivo expansion. We got that part too. We take the tumor, put it in some kind of expansion type equipment. They're saying that this process removes the TIL, which they call tumor infiltrating lymphocytes. You mostly think of T cells. Removes TIL from the hostile tumor microenvironment. So there's a tumor microenvironment sort of around the tumor and in the tumor that's a little different from outside the tumor. People have studied the acidity of this microenvironment, its predilection for glycolysis and other things. But personally, and there's also other structures around the tumor, like stroma, extracellular matrix structures. But in any event, they say extraction of a fragment of the tumor and expansion removes the TIL from the tumor microenvironment. That makes sense because you're not taking the whole tumor with you, right? You're just taking a piece. And those TILs no longer have to deal with the hostile tumor microenvironment that's fighting them, right? The tumors evolve mechanisms to fight your body's immune system. So instead, once you take the tumor fragment away, it can't rely on the rest of its buddies, the rest of the microenvironment, to stop the T cells. Well, that's important. Okay, so by doing that, it reduces the immunosuppressive effects of intratumoral regulatory T cells. So immunosuppression means less immune system. Suppress the immune system. But that's what's fighting the tumor. So you don't want that. But your body does that. Part of that is because the tumor is putting out these regulatory T cells to stop you from fighting the tumor. And they're saying that when you take the tumor out and put it in this expansion device, you'll have a lot less of that. That's great. So it says here, expansion of these outside of the body, ex vivo, rejuvenates the cells, yielding billions of such cells to be infused back into the patient. Well, again, I mentioned that there are 5 billion to 24 billion lymphocytes, period. So it's exciting that they're saying they're going to yield billions of cells, because that's kind of the amount we need to make a difference. You can't just give the guy 1 million cells. It's going to have to be a very large amount. Okay, so far so good, right? That wasn't that hard. Melanoma is characterized by a high mutational burden. So in melanoma, there's a lot of mutations in different genes. That's not always the case in cancer, by the way. And highly individualized neoantigens. What does that mean? Well, neo means new. And an antigen is something that your immune system is supposed to target. Antigenicity is what you want in a vaccine, for example. You're introducing an antigen, and you're hoping the body makes an immune response to the antigen. And in this case, in fact, we probably all have antigens and neoantigens from mutations, but because we're under a certain age, our immune system is still vibrant and still stopping these tumors from growing. As we get older, our immune system weakens, and these neoantigens start to become cancer. So when it says highly individualized, that means that there are diseases like non-small cell lung cancer where there's a very specific gene and a very specific mutation, down to the amino acid, that you can individually treat. That's not the case in melanoma, where you have a smorgasbord, a cornucopia of tumors, tumor mutations. And it's hard to sort of decide, well, what drug do I give for that? So a cellular therapy product that can address the broad nature of neoantigens and the unique array from each patient would lead to the possibility of a tailored response. Okay. Old brand name used to be, the code name used to be LN144, and we want to search for that too. If we only search for lephalocell, we may not get anything, but we need to search for this term too. And the search engines have gotten a little bit better about that. So lephalocell is an autologous TIL therapy that uses tumor tissue T cells capable of recognizing tumor antigens. That's the ideal. And being expanded ex vivo while maintaining the heterogeneous repertoire of T cells. There's lots of different kinds of T cells. Using a centralized manufacturing process. I guess what they're saying is that you don't want the manufacturing process to give you this one type of T cell, this homogeneous T cell. All right, we report the safety and efficacy of lephalocell, a one-time cellular therapy.